have Dr. Mark Goldberg with us today. He is the research thrust lead for the Matrix AI Consortium's Augmenting Human Capabilities Thrust, and he's been an active member and a proponent for interdisciplinary research in the Matrix AI Consortium. In his current role, he's an Associate Vice President for Strategic Research Initiatives in the Office of Vice President for Research at UT Health San Antonio, where he supports promotion of interdisciplinary research opportunities and a lot of external research partnerships. He's a professor of neurology and a neuroscientist with special interest in injury and recovery after stroke. He's also been a member of several other programs and centers such as the Glenn Biggs Institute for Alzheimer's and Neurodegenerative Diseases, the Texas Lone Star Stroke Consortium, and amongst others. So it's an honor to have Dr. Goldberg here, join us here and share his research on mapping the stroke. We're truly looking forward to this discussion, Mark.
Oh, thank you so much. And I really appreciate the invitation, and I appreciate so much the opportunity to be part of this group about which I know almost nothing. And so you'll forgive my my lack of knowledge of artificial intelligence, but I think that everything that you're doing as a group is so exciting, and I'm really delighted to be able to participate and to see if we can add, through our experience with clinical work or with other kinds of biosciences, to see how we can contribute to this awesome group. My topic is mapping the stroke reconnectome. And I'm going to talk to you a little bit about what stroke is and why I'm interested in how things are connected up. And I'm going to use this as an opportunity to actually present some of the technical capabilities that we've developed with a team of really awesome people that I work with at UT Southwestern. I moved here about 1.5 years ago. This team is mostly still there. And we developed a team around a particular kind of microscope that takes images of mouse brains in 3 dimensions. And we realized that once we had that, we just didn't have nearly enough to go with. We had beautiful pictures, but we did not have a way of getting data. So I'm going to show you the steps that we've taken to go from an interesting question in biology, I think, to an interesting technology question, and show how the technology is advancing really rapidly, the science behind it. So I am quite open to questions or comments as we go along, if you have some that really fit with the moment. But someone will have to let me know if they come, because I'm not sure I can see the chat. All right, so in the usual way, it's appropriate to say a few words about what stroke is. So a stroke is a sudden disruption of blood flow to the brain, which results in a neurologic impairment. And so typically you have a blood clot which lands in one of the arteries inside the brain. And if that blood clot is not relieved within minutes or hours, the tissue that would normally receive oxygen and glucose will die. The brain tissue will die, and we don't get neurons back for the most part. And so that tissue, the area of the brain which is affected by the lack of blood flow, will be injured. Sometimes, a minority of cases, what you have is not a clot within the artery, but a burst artery, which would be a hemorrhage. Those are both technically called strokes. And the impact to the person who has a stroke depends entirely on where this is in the brain. It might be entirely silent, or it may land right in the center of the part of your brain that you use for language, or the part you use for moving your arms or emotion or something like that. And so the impact for you, if you have a stroke, is going to depend on where this is, how long there is not enough blood flow, and a little bit on your own anatomy and whether you've had prior strokes. So this is a big problem. 700,000 people in the United States who have it. It's the leading cause of adult disability and second leading cause of dementia after Alzheimer's. So this is a significant clinical problem. We've had some, and I'm a part-time stroke neurologist, so I take care of patients who come to the hospital with stroke. And we've had some really fine successes over the last several years, many years. First was the advocate of this clot-busting drug, tissue plasminogen activator, TPA. And if this is infused in the bloodstream within 3 or maybe 4.5 hours after the onset of stroke, it can reduce to a significant extent the disability from stroke. But even better, a much more recent discovery is the fact that this the clots can be removed, actually. And this is a picture of a catheter which has been placed up through the leg, through the aorta, up to the carotid artery, and into the middle cerebral artery where that clot is in the schematic to your left. And a flexible metal stent has uncoiled, has opened up, grabbed the clot within it, and then it is very carefully sucked out through the catheter and goes away. Well, this is quite an amazing fact. And while we've had treatments like this, we have finally developed the ability to carefully select which patients will benefit. This is an amazing story because it actually comes back to AI. There's an AI application which was tested along with this treatment, and it allows us to see which patients have the right kind of, they have a perfusion deficit, but the tissue is not dead yet. They have the ability to benefit from this. And so in everyday usage now, we have an AI app that we use on our phones, and it comes from the CT scanner, and it tells us the data we need to make a decision about whether this device will be effective or not. This is a great story for another time. So just to show you in numerically, if you judge the success of a treatment by the number of people who are independent afterwards, the people who get only TPA, which is better than just only placebo, additional 25% of people will become independent who would not otherwise be with this clot retriever. So those who can benefit from it, it's a tremendous advance in our field. However, that's not the whole story. The story really is today about disability and deficits and recovery. The good news is that just about everybody has a substantial recovery after having a stroke. Now, that extended recovery depends very much on how severe the stroke was in the first place. So this particular score is a motor functions test with 100 being perfect. And if you have a very mild stroke, you're very likely to have a full recovery. But if you have a very severe stroke, you only have a partial recovery. This is statistically on the averages. But what's really interesting and informative is that most of this recovery happens in the first 30 days, and by 90 days, it's kind of plateaued. So what we know about stroke recovery is it's time dependent, and so that's interesting. There's a critical window for the recovery. There was a question about a critical window for how soon we could administer the clot retrieval. Well, we thought it was within 3 or 4 hours, just like it is for this TPA drug, but it turns out that every patient is different, and the tool that we have now that allows us to tell whether there's a deficit in perfusion and also whether the tissue is dead allows us to have the option, the potential that some people may be able to be treated up to 24 hours after the onset of the stroke. Unfortunately, it's not most people, but we're able to find people like that because we run a quick CT application that shows that while there's a large area of tissue that's not perfused, it's not dead yet and therefore would benefit. I hope that answers that question. So this interesting window, there's 3 really important facts. Most people recover. The recovery is partial, and it's concentrated very much in the first 30 to 90 days after stroke. Gives us hope that we could understand what is this window of opportunity, and how can we promote this? How can we get the brain to do better what it does already in terms of recovery? Okay, so briefly what we know about the field of stroke. This is your stroke 101. I'm going to tell you everything you need to know. That we've had real advances in stroke, not just in this acute care I told you, but also in prevention. So there's people are having stroke at later ages because treatment for high blood pressure, smoking, and cholesterol exists. We have improved this acute care that I mentioned, and also the systems of care, so that if someone calls an ambulance, they have a very good chance of going to a hospital which can take care of them. So these things have all improved. However, even with the best treatments we have, they only get a small portion of the patients, and so many people still have a significant disability. And as a result, stroke is far and away the #1 cause of adult disability in the United States and globally, and there's no drug treatment to improve functional outcomes in stroke survivors, although there are some devices that work. So this is the big concept for the day, is that recovery from stroke involves activation of new brain areas. It does not involve growing back tissue which has been killed, okay? And this is This is an old study, but it's sort of a pivotal one. It's from 2000, and it's a PET study, positron emission tomography study. And basically, it shows in colors areas of brain activation when you do a task. This is a language task. The person's being asked to speak, and they would do so, but they've had a stroke in the language area. And you can see there's a hole here. In this particular scan, the left side is on your left, and that's the dominant area for language. And this person can't talk really because they have lost Broca's area, for example, the area where you would produce speech. And yet something interesting is going on, which is the brain is activated just the same, and they're actually able to produce some words. And there's some activation right here next to the lesion, the perilesion area, and there's lots of activation on the opposite side and even other places. This is very interesting. Something is happening here that just this one task, the brain is recovering by assigning other parts of the brain to the same task that otherwise would never, ever be activated. And I'll show you this picture sort of done in fMRI world, so a little bit more contemporary MRI kind of techniques. And this is a similar, I think this is actually someone moving their hand. And what they're showing is the area you normally would use to move your right hand. And that has been lost in this particular patient, most of it. And now you see all these other areas, including the opposite side, being activated as the patient recovers. There was a question, are there consistent patterns? And there are. And in fact, they're kind of shown here. There's a lot of variability in this. And it seems very much that the less damage you have, the more likely you are to activate in the perilesional area, around the area of the primary thing that you've lost. Whereas if it's been a very severe injury, you're more likely to involve the opposite side. We have people who had strokes as babies and it was never known. They have no left hemisphere, quite amazing. Sometimes we'll see patients in the hospital and discover by accident that this happened. And their entire language area is gone, but they have normal language. And their entire motor area, which should be controlling the right hand, is gone. And they're just moving both hands just fine. And clearly, the ability of that other side of the brain to take over has worked for those people, and it certainly is true in children. So there are patterns, and understanding these patterns is kind of the goal of the body of research that we're working on. Okay, so here's the summary up to now. The thing we're discussing is plasticity. So plasticity is the ability of neurons to change their activity depending on things which have changed in experience or environment. Okay, that's plasticity. Learning is plasticity. Learning to ride a bike is plasticity. Forgetting is plasticity. Any kind of change in the nervous system that results from experience you have is plasticity of the brain. And so our brain is very, very plastic all the time. And what I'm telling you is that functional recovery after injury requires plasticity of the brain. We do not get cells back. The spontaneous plasticity is absolutely the best in younger people and animals. It's absolutely the best, as I showed you, shortly after injury, and it seems to, and it makes sense, seems to be strongly associated with doing activities. I mean, if you want to learn to play piano again, you're going to have to play piano. So practice specifically devoted to that activity seems to be important. And we really think that understanding the mechanism of this kind of plasticity may be useful for directing new therapies for recovery. And it's a great time to be in my field because plasticity is everything neuroscientists do now. And so we have amazing tools to study plasticity by application for the other kinds of plasticity that occur in learning and in forgetting and in development. So there's a lot known about brain plasticity, and so we have the opportunity to take advantage of that knowledge. So in the most general way, there's a couple ways you could think of plasticity. One is you could take all the synapses that you have already, all the connections there are, and just tweak the strength of the synapses. Okay, pretty sure that's how neural networks work. Don't quote me. And you have all the connections, and you could very rapidly up-regulate some and down-regulate others. And so that is a kind of synaptic plasticity, which happens on a second-by-second basis. It's pretty fast. Another kind of plasticity, circuit plasticity or structural plasticity, is to physically make new wires and new connections. I'm going to be talking about that one because we have some evidence that particular one applies in one of our projects. So I'm going to be talking entirely about changing the structure, changing axons, and creating new circuits that weren't there before. But ultimately, all that plasticity doesn't mean anything unless you have behavioral plasticity. You've got to be able to do a thing. And so it's important to remember that the plasticity might be bad. You might end up with worse function as your brain adapts to change. And that can happen in a lot of ways. Seizures can happen, or pain, or spasticity. So definitely plasticity by itself is not defined to be good. You need to have the plasticity which leads to behavioral recovery. It has to be restorative plasticity. And I'm going to go ahead and talk about the work from our lab. So I want to start by acknowledging all these people. A lot of this work was done at UT Southwestern, and we still have a lab and a group there. The folks in yellow are grad students and a former trainee who did most of the work here. And this is Denise Ramirez and Julian Meeks, who have been leading the project on our data analysis pipeline. And we've had a lot of support from a lot of people going along the way. Okay, so if we're going to study something, we have to choose a model. You always got to choose a model, and we like mice because there's a lot of good tools with mice, and there's a lot of genetic things, and we know a lot about mouse brain anatomy, and we want a behavior that seems to parallel what we do and what mice do. And you know, mice talk to each other, but not the way we do, and they run around, but not the way we do. But one thing they do, which seems to be very similar to us, is they use their hands a lot, and they use it for kind of the same reason I do, which is to get food to my mouth. And so this is sort of just illustrating they're pretty dexterous. They actually have fine motor coordination, and so we have chosen forelimb, paw, motor, meaning movement control, as the model system that we're going to work on. Okay, we know, first of all, we can give it a stroke, we can modulate this, and we've tuned our behavioral assay and our anatomy and our stroke all to the same behavioral situation, which in this case is reaching and grasping. Everybody good with that? That's an important thing. You're choosing a model, you got to explain why this is our model. Okay, all right. So I'm just going to go ahead and tell you what our model is and how we do it. So we do give the mice a stroke. We don't do it in the regular way. We don't block an artery directly. And the reason for that is that we really want to be sure that the stroke is exactly in the primary motor cortex. And there's a technique that works for that. We inject a photosensitive dye called rose bengal into the bloodstream, and then we shine a light through the skull. We don't open the skull, right onto exactly where we know the primary motor cortex is going to be. And if you check back in a few days, although this is actually our colleague's paper, but you'll see that, in fact, a beautifully carved out stroke, which is pretty consistent from animal to animal, exactly gets the primary motor cortex. And actually, I know that it is so, and I'll show it to you in a minute, because that's where the neurons are, which connect to the paw. We also have a behavioral test. And this was developed by April Becker, and who's at University of North Texas, and Dee Betts, who's an MD PhD student here. And the mouse has to, it's an isometric reach task. They reach for the thing and they pull it. They don't actually, it doesn't actually move, but they've been trained, they know. If they do this, if they pull, they get something that is better for them than any other stimulus we found, and that is a droplet of peanut oil. They like peanut butter too, but we couldn't push peanut butter through the pump. So peanut oil, they like that. And basically, if they pull, we can see how hard they pull, how fast they pull, how often they pull. We get a large amount of data, and this system will also train the mice to do the task. So it's an automated task, a little bit different from what you saw first with the rat reaching for the pellet, because that requires someone to put the pellet there and some complicated video analysis. In this case, our behavioral assay is matched to what the stroke is. And we know that because, and this is not the best data, after a stroke, these guys have continuous deficits. A lot of tasks that you give a mouse, they can recover after a stroke pretty fast because they don't use their paw, they don't use their primary motor cortex for everything. but this particular task they do. And so there is a deficit which persists for weeks and weeks out. And that's really what you want if you want to study how to make things better. Questions? And now I want to tell you why I think there is a particular kind of plasticity in the brain. And I have to explain this, and it's a little bit complicated. We know where the primary motor cortex is, I just showed you, and we can place a tracer, not on, let me just back up here. We can put a tracer on the opposite side, on the contralesional primary cortex, which is not injured, and that tracer will fill the corticospinal neurons. And what those neurons do is they go down, they cross around the level of the spinal cord, and they travel down the opposite side, and they supply the opposite side of the spinal cord. And when you do that tracer, you can see them all filling here on the opposite side of the spinal cord, and very, 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 very few axons on the side of the spinal cord that they start out. Mice are almost 100% crossed. People are 90% crossed, but mice are almost 100% crossed, which makes our work easier. And what we found some years ago, and other labs know this very well too, that after a stroke, some axons cross over to here. And about 3 times more axons, 2.5, 2 times more axons cross over after a stroke. Normally, it just doesn't happen. So the axons which belong on this side cross over, and that's good because they're there to make synapses with the neurons in the spinal cord which have lost their control, have lost their connections. And so this is a sort of schematic of what we think is happening. These little green things, after 2 to 3 weeks, they sprout and they cross the midline. This is not okay. They're not supposed to do that, okay? This is not a normal thing for an adult mouse to do. That happens maybe in development, but it doesn't happen in an adult mouse. And what's weird is these guys here, they weren't injured. They're not close to the injury, but somehow they get the message, it would be really good to go over and help out your friends on the other side. This is like the basic paradigm. How does that happen? And what can we do to make it happen more? Okay. So this takes a few weeks, and other labs have actually shown that this is definitely part of recovery, because after the animal has recovered, if they block just that pathway, the mice lose their recovery. So while there's plenty of things happening all over the brain after stroke that is all plasticity, at least this particular pathway in the mouse seems to be important, because if you block it, you lose what you gained before. So this is the model and the topic for the entire rest of the talk here. Okay. Any questions about this?
There's a question in the chat, I think, from Gabriella.
Did I answer it maybe?
It was answered. It was answered. I was asking how much time after the stroke, how much time? You said it, in a few weeks.
Yeah, it takes a few weeks, but within a few days, we can start to see inflammation in the side of the tract that's injured, okay, which is what you'd expect. The axons down there in the spinal cord, even though they're a long, long way from the stroke, they know that something bad has happened, and you start to see some inflammation there, microglia. But it takes 2 weeks. By 4 weeks, you have the most axons, and by 6 weeks, it may be a little bit less. So it takes a few weeks, and it kind of tracks with what we know about behavioral recovery. So I'm going to do 2 little bitty science stories around this just to show you why we care, and then I'm going to talk to you about the imaging pipeline we use to understand all this. Okay, so the first one is from Amy Chen, who's now faculty at the University of Kentucky, and she's interested in how glia regulate this injury response. And she starts with this observation. I think we know in the brain we have neurons, and we also have astrocytes, which are star-shaped cells, which have been called supporting cells of the brain. There's way more astrocytes than there are neurons, and they're not supporting cells. They're quite active in maintaining the environment for the neurons. But sometimes when there's an injury, if there's a trauma to the head or an infection, the astrocytes do something different. They become reactive. They take on different forms, and sometimes they will form actually a scar And if you're having an infection, that's actually a pretty good thing to have as a scar on there. But we never know whether these reactive astrocytes are healthy or are they harmful, because they're there to, they're there to wall off a lesion, but what are they doing to stroke recovery? So Amy asked this question in a particularly novel way. She discovered that there is a regulator of this astrocyte reactivity. It's called LZK, which she found out. And she found out that if she could make genetic mice that overexpress LZK, and it's done conditionally, so it happens, you control it with giving a drug. Those mice have wildly enhanced reactivity in their astrocytes. So this is the normal mouse with GFAP as an astrocyte marker, and this is what it looks like after the overexpression of LZK, and this is what it looks like in the brain. And she did experiments on this in spinal cord injury, but we're going to do stroke, and this is just published in the last week. And what she did is the same experiment that I've been telling you about, and it has the same rules that I told you, which is all of the axons are on the one side where you'd expect them to be, and almost none on the other side. And when you make the overexpressing mouse, that doesn't change. It doesn't, you don't get a lot more because of that. In fact, it doesn't really change at all. After a stroke, as I told you, in the normal mouse, there's more axons over here than there used to be, because that's part of the recovery from stroke. But in these mice that have the overexpressing, that have all the astrocyte reactivity, there's a ton more axons, much more than just stroke alone. And so we take that there's something about this astrocyte reactivity which is actually promoting this. Now, we don't know exactly what it is, but we noticed that those astrocytes excrete a substance called cellular CNTF, which is a growth factor. So perhaps that growth factor they make is helping the axons come find the right place or become eager to move ahead. But there's some signaling going on, and this is just an example of the kind of experiment that we can look at, looking at the environment of the spinal cord and understanding what promotes this kind of axon to cross. So that's a very brief version of this one paper, which is just out. And Katie, okay, so the question, thank you. My problem is it goes away before I can get to the end of it. So it sort of shows up on my screen, and then all I saw was, does this mean you can force recovery? I would like to think yes, but that seems too simple. So it seems to me that we can maybe force a state which is more plastic. And the bias on everyone's part is that that would only be affected if you pair it together with the right kind of activity. So I'm hoping that you can force a hyperplastic state, and how you would do that is still to be discovered. One of my colleagues 6 or 7 years ago did an experiment where they gave a mouse a stroke and they didn't see a lot of recovery because they didn't give them any training. And then they gave them another stroke and gave them training this time, and they got the mice to go all the way back to full recovery. And so, yes, but we're not doing this in the clinic right now. We're not giving people the second stroke to see if that might help. But the concept is maybe the hyperplastic state, could be, this is just evidence that you can recreate that hyperplastic state, which is what we want.
Thank you.
Yeah, so Katie is a PhD grad student who's still at Southwestern in our lab, and she has been particularly focused on these cells called microglia, which play a very interesting role. For a while, it was thought that the brain didn't have inflammatory cells or immune cells, and it turned out that the microglia are those, and they are very interesting. They are sometimes cause inflammation and degeneration, and sometimes they promote regeneration. And so she wanted to figure out which of those it is. And there's just every evidence that microglia could be good to support plasticity, or it could be bad. And it obviously depends on the situation. So she took it on to study this. One of the ways she did was to look in the areas where all these axons are And she found that the denervated side has a dramatic change in the morphology of the microglia, and the microglia seem to be engulfing the synapses. That's really interesting. She looked at this in a very quantitative way. Not all the synapses, but the ones that are coming from the other side, right? And does this mean that that the microglia are helping them, they're supporting them, which it might be? Does it mean that it's getting rid of the synapses that aren't needed, which microglia would be good at? We don't know what it means yet, and she's doing a very fancy experiment now to specifically knock out the microglia at just the right moment and see what happens here. Oh, well, we don't need to see that. Okay, so this little science diversion is to say that we certainly believe that the glial environment has a lot to do with this axon plasticity after stroke, and both astrocytes and microglia and possibly other cells can clearly participate in an active way in what's going on. But we also think that there's changes in the neurons themselves. If they're going to create these sprouts, and if those sprouts are going to be doing things that they normally would never do, there's going to have to be changes in the gene program that the neurons are experiencing too. And Deanie Betts, who's in our lab now, is very focused on a particular set of master regulatory transcription factors, which might be regulating that neuron change. So we'll see if that's true. We're just starting with that. Okay, so now I want to tell you about the techniques we have started to understand to use to get big data from little brains. That's the goal here, okay? And so I'm calling it mapping the stroke. Oops, well, it's going to be the reconnectome, and I'll tell you what that means in just a minute. But really, I'm going to be telling you about a set of projects which involve three-dimensional microscopy, and then what we've had to do to get these ginormous data sets to become data and not just pretty pictures. Okay, so in omics is the full set in biology of all the genes or all the proteins or whatever. And so one of my colleagues years ago invented the concept connectome, which is a comprehensive collection of all circuit connections in the brain. And we are not close to knowing what the connectome is, even for one tiny part of the brain. And so I thought, okay, that's cute. Why don't we call ours the reconnectome, which would be a comprehensive collection of the circuit connections after brain injury or recovery. And then we could compare the original connectome with the reconnectome, and we could figure out what facets of the reconnectome are reparative and what facets are harmful. And just like you might do proteomics, you might look and say, after something has happened, there's a whole bunch of proteins which are upregulated and a whole bunch of proteins which are downregulated. But that's not enough. You still have to figure out which of the ones are important and what are they doing. But this is a very good starting point. And so the reconnectome is a concept here to say, could we endeavor to understand what connections have changed using the tools we have? My hypothesis is that the new connections are responsible for some of the functional improvements that we see after injury. Okay, so turns out we have some ways of seeing axons in the brain, and all the brains I'm going to show you are not live brains. These are all mouse brains which mice have been sacrificed, the brains have been fixed, and they're looking at the microscope. None of this is live imaging, although that is a very cool and interesting field. So this is an example. Some years ago, when mice with green fluorescent proteins were beginning to first be engineered in the brain, our colleagues invented a series of these things, and one of the lines of mice that they invented turned out to just beautifully label axons in the brain. So it turns out we can actually see axons really well. And I thought, okay, now we're good. We can know how everything connects. We've got our problem is solved. Everything we need to know is known now. But you only have to look a little bit closer to realize that there's way too much information here. If everything's traveling in the same direction, you're good, but not when they're crossing. And so you can sort of get the idea of where the tracks are, but the reality is that this kind of imaging gives you a nice image about whether the axons are injured, actually. And so we can see all this injury here. So it's really good for that, but it's not a good way of tracing all the wiring in your brain, okay? And it's no different than if you were to take your circuit box in your house and say, okay, I'm going to figure out how everything works. I'm just going to pull off the the breaker box and look at the wires inside and everything will be known to me. It's not. So we need some other way to understand the connectome. So this is a technique, this is a process that we did some years ago. And it wasn't perfect, but it works. And we injected a retrograde traveling virus, one that's like rabies, but it's pseudo-rabies, into the muscles of the forelimb of a mouse, okay, the front paw. And this particular virus, like rabies does also, travels back into the brain. This is how rabies is so bad. It goes through the muscle, through the nerve. It comes to the first neuron in the spinal cord, and it synapses onto the next one and goes up to the brain. And this is how I know that the area I showed you is the primary motor cortex, because it has all the connections. These neurons are all there. Okay? But there's a lot of other areas. There are some on the other side. And the challenge with this are two things. One is it turned out this particular virus was toxic and was killing some neurons. And the other is we don't know if we're looking at the first order connection or the second order connection or the third order connection. We don't know if these guys were directly connected to that paw or whether they first came here and then snapped there. And so that was a challenge to understand, but it was a really good model for which to begin our imaging. So, and I have to just tell you, there's a quick diversion. There, when you're doing microscopy and imaging in general, you got to know what scale you're looking at. So at the electron microscope level, you can see individual synapses and dendrites, individual ones, but you can't see what they're connected to because you only have the little teeny field of view you want. At the macro scale, for example, MRI of the human brain, you can see the pathways, but you are never going to see the individual cells that they come from. So this concept of mesoscale was kind of invented, and I'm going to be showing you mesoscale microscopy, which is we're getting cellular level resolution from the microscope, but we're collecting all of the data all around it. Yeah? A little like Google Maps, or Google Earth rather, for the brain. And this is the device that we're using to accomplish that. This is called serial 2-photon tomography, and this machine is called the TissueSight. And basically, the fixed brain is placed on the stage right there, and a multi-photon microscope takes a series of pictures. I better play it again. And each time you're doing that, that's one whole field of multi-photon beautiful images. And when it's finished taking all those, the brain stays right where it is, and the vibratome comes and slices off the part you've already imaged and discards it. Okay, what's good about this is this keeps the three-dimensional structure. If you tried slicing all of the tissue onto slides and then reassembling it, it's really difficult to get this reassembled. Okay, so here, a long ago picture of Julian Meeks and Denise Ramirez looking excited to do this experiment for some publicity photo. But they're the ones who, in fact, set this up. Okay, so this is serial 2-photon tomography. These days, light sheet microscopy does a similar thing. It does three-dimensional imaging through an entire mouse brain without having to cut the brain up to get the pieces. All right, so in order to make this work, you need a kind of, there are steps here. The last step was image acquisition. Now it turns out you've got many, many, many individual pictures that need to be stitched together. There's an auto stitcher in ways that you don't see the lines in between. And then you need to have massive amounts of storage, because when this is done, it's going to be quite a large thing. So let me just show you what we've got here. This is one picture, one field from one image here from the experiment I showed you before. And that's just one of a great many that get stitched together to make a plane like this. And now it tells us some good information, because we can see sort of how these areas are related to each other. But if we want the whole thing, we need 3 dimensions. And so to get that, we need to have a whole bunch of these planes and put them together like so. So that was the work of quite some time to make this all happen. And it works. And so we can actually see the entire connectome, all the neurons which have a connection to that front paw are captured here. Many, many thousands of them, or more, I don't know, a large number. But there's a problem, which is now what? We can see this, but we don't know exactly how to analyze it. We have 70 gigabytes of data. It took 10 hours to image, which is incredibly fast. And what we get is a whole series of beautiful little rotating brains. Okay, so we were just really awesomely great at making these kind of rotating brains, and you can see that there's a lot of neurons in here, but that's hard to generate information from that. And here you can actually, we have the ability to, in transgenic mice, to have two colors, and so now you can see, you know, connections between two areas. And there's a lot to be learned here, but at this, it's very difficult to make sense of this at the cellular level or to get quantitative data out of this. We did eventually. And the thing is, what you'd love to be able to do is you could do on Google Earth is zoom in and say, I'm really interested in this area of the striatum. Show me the cellular resolution data, which is still there. It's in the data set, but it's such a giant data set that we haven't been able to do it. In a couple instances, we have used Imaris software to program a fly through. Oh, here's our stroke reconnectome data. Like I said, it's all there. It's not possible for us to understand it. So we did make one, and if you're seasick, you might want to look away. But this is a kind of a fly through of the data, which shows the ability to, the fact that the cellular resolution is actually in the data. You just can't see it unless you get close. And so we don't have the ability to zoom in and see these things. I've tried virtual reality. I've tried projecting these things on a planetarium ceiling. But you know, ultimately, this is a data challenge, which is how to be able to keep and hold a lot of data and also be able to see subsets at the same time. So our challenge is still how to visualize the data and really understand it well. But the bigger challenge is how to understand it in a quantitative way. So for that, we have a whole set of steps, and one of them turns out to be very convenient. It is possible to do, and this is something you do in the world of human MRI and all the time now, register our mouse data into an atlas space. The Allen Institute, which uses similar techniques to us, have created a common coordinate framework, which they did by averaging 1,600 mouse brains. And what they did is they took the channel, which wasn't labeled, just the autofluorescence, and they got enough information here to beautifully sort of capture the average mouse brain. And it's so highly resolved that you can even see what is called the whisker barrels. These are actually the areas of the brain in which each whisker connects to. And so this is something you can't, looking at a mouse brain, you would not be able to see with your naked eye, but it's in the data. So this is a very highly connected set, and it is possible for us to use software to register each of our brains into this common atlas space. And so this is an example where the little cells which have been labeled, or the stroke, is now going into not the space of the mouse that we image, which might be tilted or might not be exactly the same, might be a little bit bigger, might be a little bit smaller, but is being registered into the actual space of the reference atlas. So this is a step towards being able to understand the data. And the last thing you need, oh, two more things. This was the easiest, the artificial intelligence piece of this was far and away the easiest piece. So our colleagues used Elastic, which is a voxel-wise random forest model-based machine learning paradigm, which involves the user, any of us, going through a sample data set and scoring the features of interest. In this case, we wanted to know, is it a neuron, is it a dendrite? But sometimes we want to know, is it a glial cell, is it amyloid? And we simply score it enough times, as in any kind of machine learning, until the machine is accurately recognizing. In this case, the red dots are neurons, the blue dots are dendrites, and the yellow dots are stroke. Okay, and this lives on that. It lives on top of that registry, that registered atlas that we showed you before. And so this is the projection of all of the, and then you'd only have to teach it once, and then it can go all the brains and it can count thousands and thousands and thousands of neurons and put them anatomically where they need to be. Okay, so this would be the work of a grad student. This would be months and months and months of work. They probably wouldn't do it, although we did one. to verify that the technique worked, and that person is still in science, Deanie. So, but it's not the sort of thing that you would want to do all the time, because just the sheer quantities of data this is doing. It does it very fast and it's very accurate. And the last piece you need is you need to know where the brain regions are. And in this case, we have the advantage that that's been provided for us by the Allen Institute. And so what they've done is they've created an annotated atlas where each of these areas is a recognized part of the brain with a name and a number. And so the system can then take all of the classifiers that we did, and what it does is for each region, it calculates the average probability that there is a feature that you want, that there's a neuron there, or not, whatever the thing is. It gives you the probability map that that region, that 3D region, has that feature in it, that this region has that feature in it. And you can see these are obviously arbitrary, but they're the classifiers. And with this, you can now say, for that connectomic atlas I gave you, we can tell you exactly which neurons are in which areas. And that gets a heat map, which is a sort of map you get with omics, where the color of the line tells you the intensity or the probability for each of these, I forget, 200 something, 532 independent brain regions. And each of these is a mouse, and the left hemisphere and the right hemisphere. So from this, it is possible to generate large quantitative data sets in which all of the data have been allocated to a spot, a region. We have confidence when the regions are big. When the regions are very small, you have to worry that the classification or the segmentation hasn't been right. And that's something we'd like to talk to you about, because we're relying on the atlas space, and the atlas space just might not be that accurate when you're looking at little teeny brain areas. And this is particularly true in the spinal cord. But for big areas, primary motor cortex, that kind of thing, I think we have a lot of confidence that this is real. This is the sort of data we get in that first set of experiments where you're measuring the number. This is, actually, this one looks like the one that was done by hand, but essentially it's the same sort of result. After the stroke, you've lost all the neurons on the side that had the stroke, but you've gained some neurons after 21 days on the other side. So there's more links on the opposite hemisphere, and that was the intent to show that here. Same thing for some other areas. So this is the total pipeline. There's raw data collected in usually a couple different colors. A classification step, which is done by machine learning. There is an atlas, and that atlas has regions that have been identified by someone else through an atlas. And then this is all merged to generate the quantitation or the probability map that each of these regions has the feature that you're interested in. So I've described a pipeline that is good for 3D visualization, but by itself does not generate any facts that we can work with. And then with image registration, data segmentation, machine learning analysis, we can get actual quantitative data. It does not have to be neurons. We've counted how many lymphocytes appear in different brain regions. It can be just count anything you like of a very large scale from a very small brain. So, and we are working to use this, but we haven't quite yet, to solve the stroke connectome, and I'll show you a different approach we're taking. Questions about this pipeline?
So it seems like, Mark, getting the data from the atlas and trying to map through each of those regions, yeah, and identifying at the neuronal level if it has the features that you're looking for seems to be quite time consuming. Just thinking.
Oh, no, it's incredibly fast. If we do this by hand, it's unbelievably time consuming. This is very fast. So the machine learning, you have to train the machine. That's a matter of just a few hours. Yeah, it's a few hours. And once you've done that training, that algorithm is going to work on all the brains in your series. So extremely quick. You do, one round of this, you verify to yourself that the next 50 or so areas have been done correctly, and you walk away. So this is indispensably fast. I mean, it's incredibly fast. The allocation to brain regions is also done it's, it's a high performance computing center, and so it's done in a matter of hours, and that's fine. The challenge we have is that we don't trust the segmentation when it gets small. And in fact, we can look at the published atlases, and sometimes looking at the left side and the right side, they don't even show the same exact results. These are just such small regions that we're not sure that we can trust that this is the left side of the ventromedial hypothalamus, but we're pretty sure it's in the brainstem. Yeah, so I think that we could do better by using some intelligence to classify the regions than we have so far, and I'll show you some of those ideas at the end. But the classification of the features is accurate and fast.
Okay.
Count all the amyloid in the entire brain in a matter of hours, or all the amyloid in the blood vessels of the brain. Similar, just super fast, and we do that.
Okay, thank you.
Yeah. This is 1 to 2 days instead of months and months and months. Okay.
The smaller subsections, are you using the same, I guess, model for kind of predetermining or segmenting?
Say it one more time.
Are you using the same model for segmentation for the smaller subsections of the brain?
Well, yes. I mean, that's the challenge, right? This is the published data set. So this here. This is the data set. This is the atlas, the Allen Brain Atlas. And so if you were to go online there and you look at the serial sections and you want to find primary motor cortex, you'd find it shown there on their atlas that they already have. And so someone has had to decide manually that this area is primary motor cortex, right? And so you have to trust that segmentation was done according to neuroanatomists' best guess about what the region should be. When you start getting to these areas which have a lot of subregions and a lot of nuclei within a small distance, it seems arbitrary. And also, it feels like the distances between the groups are smaller than the confidence that we have within the region. So we would like to supplement this by using some intelligence to say, I think this is the region, and I'll show you in the spinal cord, I think this is the region where the motor neurons are, and here's how I know, okay? Meaning we could use the same techniques that you use to analyze pathology or anything else and say, based on the morphology of this blob here, I think this blob belongs to the lamina 9, okay? But right now we're not doing that. We're using the dimensions, we're using the stereotaxic atlas, which has been registered into space here. Does that answer your question? I hope. Okay, so we've started to ask the question directly, looking at the spinal cord. Okay, and this is harder, but more informative. The spinal cord is arranged in segments. So you have your cervical spinal cord, thoracic cord, lumbar cord, and sacral cord, just like mice and people have that. And at each of these levels, there is the gray matter where the motor neurons live, and they send their connections out this way to innervate the muscles. So in order for a movement to happen, the axons come down here from the brain, and they synapse either on an interneuron or directly on one of the motor neurons which live here. And that motor neuron sends a direct axon right to the muscle, only some muscles, and that muscle contraction gives you a movement. So we know that we're particularly interested in some of the interneurons which connect to motor systems, and we're interested in the motor neurons. But we're also interested in the idea that there may be pain pathways or the kind of pathways, some of which maybe the connections to would actually be harmful and might explain why you sometimes get adverse, like after a stroke, spasticity, that kind of thing. So we would love to know, when we see this kind of connectivity that I've been talking about, where are these axons going? Up to now, we simply said, look, there's more of them crossing the midline than you'd expect. We didn't ask where they were going. So we've developed a whole new, and these lamina here are just names for the areas which have been determined by other people, lamina 1 and so on, right? So these lamina are, and these names for regions are ones which have been characterized by classical neuroanatomy over the years. We'd love to know, when we see the reconnectome, where are these axons going? And, first of all, can we get more of them, but also can we be sure they're going to the right place? So for that, Katie Poinsett has been working very hard to develop a way to see exactly where the axons are going. And she has an advantage of a virus from Weizhou, our colleague at UT Southwestern. And this virus causes neurons to express in their axons a green color and yellow color, and in their synapses, presynaptic terminals, a green color. So we can see all of those things. And so normally after no stroke, you don't see anything on the other side. After 1 week of stroke, you can start to see some axons, and after 4 weeks, there's just a ton of them here, and lots of little green synaptic points of contact, lots. So we can use the same machine learning that we were telling you about, the pixel classification. There's lots of little red pixels on the side that is innervated, but also quite a few here, and we can start to map these little presynaptic sites to that atlas I showed you, except we don't have an atlas. So, but we can make a 3D picture like this, which doesn't really tell us much. And fortunately, another lab just last year did publish an annotated atlas. So they basically did exactly what the Allen Brain Atlas had done for the brain, and they just divided up these regions, and they gave us the color-coded annotations that we could put into our system. So that's what we've done. And we can compare in gray the atlas, and in red what we're actually seeing as we go from the top to the bottom of the cervical spinal cord. Okay, and it's pretty good, but I think it could be better in terms of how close is the overlay of these regions to what the actual data show. So this is where we need to work, and this is really tiny. These are teeny tiny areas. So with that, we can get some data. And so this is an example of the kind of data that really is helpful. These are different regions of the spinal cord. And just like I showed you that there were more axons traveling, there are a ton more synapses in this area and this area, area 4 and area 5, at 4 weeks, but not 1 week after the stroke. So these data here are the automated counts for this region, and the blue automated counts for all of these regions. So we've used the whole, this whole pipeline has been adapted to spinal cord using the annotated atlas that was given. Okay, I have a question, Gabriela.
So I'm curious, all of this, what other things do the patients do to trigger these behaviors? Is it all based on their genetics and their brain plasticity? Is there any external factor that they can use to trigger more? Anything?
These are mice. These are professional mice. They're mice.
Yeah, I know. What can you do? Is there anything, an external factor, anything else?
Absolutely, there absolutely is. And as soon as we know what it is, we'll share that with you. That's part of the goal here. So all the thing we know is that a certain amount of practice is very important. We don't know what kind of practice is good. We do not yet know what kind of behavior, when it's good to give it. And one of the things is if we have a really good biomarker of recovery, that would tell us. So if we had a way of seeing it here, we could easily test a transgenic mouse that overexpresses this, or we could test a certain kind of behavioral paradigm. So the goal here is to use this information to answer your question. We absolutely want to know that. I'm going to skip to...
I will follow your work, by the way. It's fascinating. Thank you
Wonderful.
so much.
Okay, well, we hope that we get some collaboration and contact here. Here's the big goal, okay? So what I showed you is a map that is defined by regions that are anatomically defined. We would like to be better, and this is what we'd like to see help. Ultimately, we want a cell type specific connection map. When we say something connects, we don't want to say it goes to lamina 5. We want to say it lands on a synapse which is cholinergic, or there's a wonderful cell specific atlas which are defined molecularly, or it's landing on the neuron which controls the biceps. Okay, so our long-term goal is to make this connectivity map filled with more information than we have now, and so we can start to compare the behavior and the connectivity map and say this side of connectivity seems to go along with good repair and good recovery. This kind of connectivity seems to be harmful. And that's the long-term goal and the kinds of things for which I am sort of eager to get help, because this kind of information at the level of the big data goes well beyond what we know how to do. But the information is all there and publicly available, and we just want to be able to see how can we start to be smart about really understanding in a deep way what the new connectome is. So I'm going to conclude, and then we have time for questions if we say we do. CNS plasticity is the key to the therapeutic advances in stroke recovery. I hope I convinced you about that. The mouse experiments show that there's significant structural plasticity in the uninjured corticospinal pathways. Now, I don't mean to say that's the only kind of structural plasticity or the only plasticity, but it happens to be a really convenient one to study because we can see it. The glial environment's obviously important, and the computational analysis of this stroke reconnectome may reveal the full set of connectivity changes which are associated with behavioral recovery, and therefore that could guide approaches to therapy, how we do it. Ultimately, you can imagine that this is not about stroke particularly, it's about neural circuits and in models, and basically you take the circuit tracers and the molecular things you know, we're using three-dimensional imaging and analysis, and then we're also looking to see, as we modulate these circuits in various ways, how does that ultimately end up with behavior that is better. And with that, I thank you for your attention and for your questions, and I really appreciate the opportunity to be part of this group.
Thank you, Mark. That's an amazing presentation. The floor is open for questions. I think some of them had to leave for work. But I think there are two things I see in your presentation, Mark. The first part you shared with us was talking about the plasticity within the CNS. Whether it's you highlighted the functional plasticity, and a lot of times in the AI models that we have today, the newer AI models, we incorporate these plasticity mechanisms, but we do not necessarily go beyond the synapses, right? You kind of emphasize the importance of astrocytes and glia. There is a discussion that has started, but just because of the complexity of implementation, we haven't started getting to that level. But I had one question in that recovery part that you showed with this plasticity. Once you have the stroke, whether it's whatever the level is, mild to severe, the recovery is not full 100% to 100%, right? There is a slight degradation in the performance.
It's about 50 to 60% improvement. So if you started with a very severe stroke, you're going to end up with, on the average, this is the average person, on the average, with quite a significant deficit. And if you started with a very mild stroke, you're very likely to improve completely. So there is sort of a little uniformity, but there's a great amount of variability between people. And so I think that we're more interested not telling people, oh, you're never going to get better. We're interested in saying, here's what you can do that might make you get better.
And in my naive way of asking here, usually, like, you know, one of the plasticity mechanisms is the neurogenesis, right? And there is understanding of how perhaps it can be done. But I think the complexity probably that you're referring to here is that the plasticity is just not tied to that one part. There are other structural plasticity mechanisms and behavioral plasticity mechanisms that all have to play up a role to get to that higher levels of recovery.
So what I'm saying is that in stroke, but in the models that we're studying, we don't think that neurogenesis in the form of neuron replacement seems to be playing a role in the recovery that we see in people. It's the amount of neurogenesis in the adult nervous system is pretty limited, and that when we've done experiments with stem cells and cell replacement, those cells very rarely become neurons, okay? So the role of neurogenesis and the role of stem cells replacement seems to be providing a trophic environment, changing the chemicals in the environment that might make recovery better, but we don't think that new neurons are going to the place in the cerebral cortex where that hole is. We know they're not because it's a hole. So in this model of injury, we don't think neurogenesis is a big part of it. That's not true for other models or other diseases where clearly neurogenesis can contribute to recovery, but not, we haven't seen it yet in the animal models. This was studied quite a lot, and almost none of the neural stem cells turned into neurons which stayed integrated with the rest of the nervous system and survived. So, it's just a really rare event.
So most of them do not survive. That's interesting to know.
In stroke. The vast majority, they could become astrocytes. I mean, so the neuro stem cells can become other cells, and they do. But there are instances in lots and lots of papers where you can show a neuron integrating with a circuit and having an axon and a dendrite, but the quantity of those things is small, and so it doesn't seem to be participating in the spontaneous recovery that we see now. That doesn't mean that someone couldn't find some way to make neurogenesis be better, but
But I think that's what's so nice about structural plasticity is that you don't need a totally new neuron to form this new connection.
Right. Thank you, Dee. That's right. What we're seeing is already available. And I forgot to say that I had some ideas about therapy for your neural networks and your neuromorphic brains. And the question is whether any of these things would be, if we can't fix people, whether we could fix some neuromorphic networks through the kind of resilience that this model shows.
Yeah, and that's why I think these questions the first half of the talk was really helpful for us who kind of heavily use these plasticity mechanisms in our neuromorphic systems.
Yeah.
And I think we perhaps give a lot more importance to neurogenesis in our models to some degree.
Yeah.
But definitely there are other plasticity mechanisms. And I don't think there are enough glia models, for example, that are incorporated because it just makes the models so complex to solve any real world task.
Yeah.
Because
Yeah.
our end goal is to solve real world tasks.
I thought the conversation about incorporating glial signaling into neuro, I don't know how to use any of this wording, but neuromorphic coding, let's say.
Yeah.
I thought that was really interesting, and it kind of made me wonder if I think about when I think about what kinds of effects the extrinsic environment has on local circuitry, I think of it as sort of introducing this basal signaling. So some kind of really consistent, maybe if you're trying to get action potentials in a particular circuit, then the extrinsic environment could sort of serve as this blanket suppressor of activity. So making every action potential a little bit more difficult to achieve. And I think that concept is really important in a lot of neural circuitry in general, where, you have certain brain regions that are constantly being inhibited by other brain regions. It's not enough to totally suppress their firing. In fact, they still fire a lot, but it serves that, whatever that sort of basal background signal is going on, that a particular circuit is receiving, seems to determine a lot of its properties. So do you feel like there's a way to incorporate that in a code?
Is a question for me, then?
Yeah.
Yeah, so right now we do not know what that external threshold is. When we have designed our neurons, we kind of say, okay, if it is above this threshold, it is going to fire. There is going to be an action potential. And we give some probabilistic nature to it and introduce some metaplasticity and other factors. But I didn't know if there was a common signal that I could use across my networks. If there is something, and how do I activate that common signal, right? When should it be triggered? If we have some information on that, I think implementation wise, it shouldn't be too hard. If there is a common signal, it is easier for us, I think. But we need to know a little bit more about how that works in some sense.
Yeah. I guess you basically have a sensor somewhere at the distal end of your system monitoring the health of the output, and that's what the astrocytes would be doing.
That's a good suggestion. Yeah.
Yeah.
Yeah, and I think there's great points in the second half of the talk, which is like, speaking to our machine learning researchers in here.
Yes.
Uh,
Yes.
I'm sure there are questions. Gabriella, did you have a question? I know you were.
I have a question about other types of conditions. For example, conditions that are triggered by viruses like myelitis. Oh, I don't know if I'm pronouncing it correctly. I know the
Yeah.
Romanian word.
Right.
So are those, they are also, the axons are affected, right? There is some impact there. Is there any connection that you can make from those types of conditions to your work, where you can inflict the same kind of movement of activity from one side to another? Maybe the spine is inflamed on one side but not on the other, and so forth. Is there any work there?
Yeah, there is. So in myelitis or multiple sclerosis or spinal cord injury, it has been really difficult to get axons to grow through the central nervous system and find targets. And it has to do with the fact that in humans and evolutionarily ever since salamanders, so 100 million years, we do not have regeneration of the central nervous system or our limbs. We don't grow our limbs back. And there's inhibitory factors within the spinal cord that prevent axons from growing back, and it just hasn't been successful. But there are dysfunctional axons oftentimes in a spinal cord injury or in MS, and sometimes they're dysfunctional because they've lost their myelin, they've lost their insulation, they're not traveling as fast as they could. And so in some cases, it looks like remyelinating them will help them become functional. And so the question is whether you can take advantage of those few surviving axons to do more things. And I believe that's exactly what happens, because people with spinal cord injury sometimes can regain function with functional electrical stimulation. There's a variety of things which can cause people who have a significant spinal injury to recover, and it doesn't seem to involve getting axons to grow freshly, because that has been really hard to do. So the best guess is it's using some of the axons, repurposing some of the axons which are there, and the functional electrical stimulation is making those circuits form. And I haven't followed the spinal cord injury literature, so I just assume that from my own biases. But I'm pretty sure, I say with some confidence, that must be the answer.
Interesting. Thank you so much.
That's my theory.
Yeah. Thank you. And I think at the beginning of this talk, you made a statement, Mark, that you don't know much about artificial intelligence, but I think you know a lot about natural intelligence. And that is really important for us as we are building these artificial intelligence systems. And I think this information is quite amazing, and the way you delivered this for our audience to consume, I think this is very helpful for us. And I'm sure there are researchers in Matrix who can help with some of the ML modeling at a lower level of abstraction at the finer granularity. So I'm looking forward to seeing those collaborations happen. Thank you.
Wonderful.
So much.
I am so looking forward to getting to know you all and to seeing how we can work together. It's couldn't be more excited. Thank you all.
Thank you.
Thank you.
Bye, everybody.
Bye.